I'm going to present you my results on multiple wavelengths excitation fluorescence spectroscopy for improving fluorophore estimation with the example of 5LA-induced PpIX in neurosurgery. So, glioma is a brain cancerous tumor consisting of two parts, the core and the margin. The core is composed of 100% tumor cells, while the margin is composed of a mix of tumor and healthy cells. Specialists usually distinguish dense margin with a high density of tumor cells from sparse margin with a low density of tumor cells. But the diffusive aspect of glioma makes its full resection really difficult. Therefore, since almost 20 years, fluorescence microscopy has shown its relevance in neuro-oncology, and 5LA-induced fluorescence of protoporphyrin IX, PpIX, is currently used through surgical microscopes in the operation theater. However, its sensitivity is still limited when applied to low-density infiltrative parts of high-grade glioma or low-grade gliomas. To overcome sensitivity issues, various 5LA-induced PpIX fluorescence spectroscopy methods have been proposed. Some tries to quantify the fluorescence, while the others study the spectral shape of the fluorescent response. Many studies on biopsies raise the presence of two states of PpIX with different fluorescence spectra, one peaking at 634 nanometers and the other one peaking at 620 nanometers. Both states have been seen in high-grade gliomas as well as in low-grade gliomas, but there is still a controversy about the presence or not of this peak at 620 nanometers. A recent 10-patient clinical trial demonstrated the relevance of the second peak of fluorescence of PpIX at 620 nanometers in terms of pathological classification. Various methods exist which try to quantify the two peaks from measurement using fitting methods. And the measurement is made at 405 nanometers, which is the gold standard for PpIX fluorescence. However, other endogenous fluorophore could play an important role in the fluorescence emission spectrum in the 600 nanometers region. To take this into consideration, the literature proposed models including different fluorophores such as lipofuscin, NADH, autofluorescence, and so on. But each added fluorophore makes the model a little bit more complex and adds another parameter to be estimated from a single measurement. Therefore, the fitting of fluorescence spectrum is multiparametric, and there is a strong a priori on other fluorophores. We propose now a method which is new for PpIX, using multiple wavelength excitation of the fluorescence based on the postulate that each of the end states of a fluorescent molecule has its own spectral response and absorption spectrum. Indeed, getting the fluorescence response of the same sample when excited at each maximum of absorption enables us to get the estimation of contribution of each state of PpIX without any a priori on other endogenous fluorescence spectra. We decided now to apply this new generic method to our case of PpIX. As you can see on figure, PpIX 620 and PpIX 634 have different absorption spectrum with a distinct maximum for each of them. The maximum of absorption of PpIX 620 is at 385 nanometers, and the one of PpIX 634 is at 405 nanometers. We will now consider these two wavelengths when applying our method to PpIX. We then would like to compare two different approaches. The first, named multi-excitation by linked solution MESOL, relies on a possible combination of the outputs of two mono-excitation models at 385 and 405 nanometers. The first mono-excitation at 405 nanometers is selected because it is a gold standard for PpIX fluorescence, and the second one is selected because it is the wavelength of maximal absorption of PpIX 620. The second approach, named multi-excitation by linked model, is multi-excitation which outputs directly the two parameters that best fits fluorescent spectra at all excitation wavelengths. To be able to have a correct comparison of the different approaches, we used a numerical phantom of fluorescence. As you can see on this slide, the phantom models the fluorescence and the detection. Indeed, the Poisson law here models the probability that a given photon reaches a detector cell, and the normal law here models all noises that are directly linked with the electronic processing of detector informations. The parameters of these noises have been estimated on the data of the 10-patient clinical trial. And the fluorescence emission is composed of three terms: PpIX 620 contribution, PpIX 634 contribution, and also a parasitic fluorophore whose impact on the estimation has to be estimated. The fluorophore fluoresces in the area of PpIX, which is the worst case for the estimation of PpIX, and we precisely decided to test this worst case here. So we decided to model the added fluorophore by a Lorentzian function, and we will study the impact of the Lorentzian function parameters lambda para, sigma para, eta, A on the estimation veracity. On this slide, we decided to show you results of the impact of the Lorentzian maximal polarization lambda para on the relative error defined in equation 1 for the healthy category. You can see plots of relative estimation error of the two mono-excitation models. The mono-excitation model at 405 nanometers estimates with highest accuracy PpIX 634 contribution, even if there is another fluorescence spectra implied. And the model of mono-excitation at 385 nanometers estimates with highest accuracy PpIX 620 contribution, even if there is other fluorescence spectra implied. For each one of the displayed curve, we notice that the error is relatively small if the parasitic fluorophore peaks outside of the PpIX area, but explodes when approaching the PpIX area. Now you can see in yellow the relative estimation error of the ME model approach. As we can see, this model's relative estimation error always stays in between the two mono-excitation ones. Because we have noticed that each mono-excitation estimates at best one of the two parameters, we decided to take PpIX 620 contribution from mono-excitation at 385 nanometers solutions and PpIX 634 contribution from the mono-excitation at 405 nanometer solutions, and combine them into the ME-SOL approach that you can see in green on the slide. On the right side, the table sums up the relative estimation error for each anatomopathological class. And we see that adding a parasitic fluorophore produces a systematic overestimation of PpIX. It can be very high, since we have at least 55% of error for the healthy category. And we are sad to say that the physical a priori induced in the ME model approach improve the robustness of the estimation less than the ME-SOL approach. So the ME-SOL approach thus seems to be the most effective in reducing the overestimation error of PpIX, which is confirmed by the table and especially the third column. On the graph, we compute the relative estimation error for various values of sigma para, which represent the full width mean height of the other endogenous fluorescence. The variation of these parameters and the values of the displayed tables confirm the previous conclusion. The ME-SOL approach reduces overestimation of PpIX. To put it in a nutshell, in the context of glioma resection surgery, the 5-LA-induced PpIX fluorescence spectroscopy is widely used since almost 20 years. To go over a lack of sensitivity between low density margin and healthy tissues in a mono-excitation wavelength approach, there is a need to have a robust estimation of PpIX inside fluorescence spectroscopy measurements. We suggest a new approach based on multi-excitation linked by solutions based on the absorption spectra of interest fluorophores. This new approach reduces overestimation of PpIX, but because of other measurement noises and non-consideration of added parasitic parts in models. We currently focus on a more complete multi-excitation wavelength model to even more robustly estimate PpIX. I especially acknowledge LabX Prime for funding my PhD, Pilot for the data they provided me on this topic, Manutech-SLEIGHT for allowing me to give this talk. And finally, I want to thank you for your attention. If you have any questions, I will be glad to answer them.